Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is March 26, 2026, and it's a special day for us. Today, all neuroscientists here at UTSA are spending the entire day attending our annual Neuroscience Symposium. In our symposia, we invite a small group of the best scientists in the country on some research topic of interest to our faculty. And if you follow the podcast, you know we have an interest in brain mechanisms of navigation, and particularly in the place cells, which are neurons located in the hippocampus that fire in relation to the location of an animal within its environment. And so today's topic is from place cells to computation, and our speakers are exploring how these cells contribute to some fascinating cognitive functions beyond just simple place and space. So the symposium was organized by our own Dr. Francesco Savelli.
Hello.
Who many of you may already be familiar with. And I'm just going to ask all the participants to introduce themselves with a name and professional affiliation and maybe a tiny thumbnail description of what we're talking about today. And I know it's impossible to fill in all the details, but the listeners will get a more satisfying idea as we go along, I think. So can we start there?
Sure. I'm Jill Loitgev, and I'm at the University of California, San Diego. And yeah, it's been a pleasure to be here today. And all the talks were dynamic. And what I talked about today was using the neural mechanisms in the hippocampus for not only where you are in terms of your sensory coding of your actual position, but using being able to explore space mentally, to have representations of space that you have been in or you're going into, and how that might inform working memory, and really focusing in on regions of the hippocampus and their contributions to those kinds of coding schemes that are used as we explore space and make decisions and navigate.
I'm Annabelle Singer. I'm the McCamish Foundation Early Career Professor at Georgia Tech and Emory. And today I talked about how we navigate in a dynamic world, how we learn where the important places are, and then how we figure out how to get to them even when our environment changes.
Hi, I'm Cameron Diba. I'm an associate professor in the Department of Anesthesiology at University of Michigan Medical School, working in studying the role of sleep in memory-related processes. So today I talked about some recent work we had that showed that after sleep, the frequency of theta is lower, suggesting it might be a signature of a well-rested brain, and some other work showing that spatial representations stabilize and retune during sleep.
Hi, I'm Albert Lee. I'm Associate Professor of Neurology at Beth Israel Deaconess Medical Center and Harvard Medical School in Boston, and also investigator at the Howard Hughes Medical Institute. And so we study, like everyone here, we study the hippocampus in rodents, in rats and mice. And lately we've been using it to study higher level cognitive phenomena, such as vivid recall of spatial, let's say, memories of places you've been to, as well as how that could lead to imagination, spatial imagination, as well as planning.
And I'm Francesco Savelli. I'm from here, UTSA, and today I talked about how possible mechanisms or how entorhinal inputs could be plastically integrated in place cells while generating a place field, and also exploratory behaviors that have been linked to the emergence of place fields in hippocampus.
So we can't reproduce the whole symposium. It would take a whole day doing that again. It would be fun, but take too long. And so I'd like to maybe suggest a couple of themes that I saw during the day. And the biggest theme I think unquestionably of all of your talks was examples of meaningful contrafactual firing of cells that would normally say where an animal is in space, but now they're representing something else, like where the animal's going, what the goal is, or maybe even, I mean, Albert showed uh, animals Jedi style moving a virtual object around in space by making place cells fire in the place where that object ought to move. And of course, it took an experimenter to make the link between the place cell firing and the movement of that object. But once that link was there, the animal would move the object just by making its place cells fire. And so that creates, I think, a kind of image. There were other things like that, maybe not quite as Star Wars, but they're the same thing. It's this place cells are representing something other than just the place of the animal. And so we're used to thinking about the number of place cells that are firing at any one moment where I am sitting here is relatively small. It's just the ones that represent me here, and the others are quiet. But this raises the possibility that if I have control of other objects in the room, that as I move them, there's groups of place cells that represent that object and other ones that represent that object, and also about goals of where we're trying to move to and where objects are moving to. And all of these things are simultaneously represented in groups of cells, in which case, experimenters might, although haven't yet in the work shown today, sort out all these different groups of cells and separate them, decode them differentially for all the different things that are going on. But presumably you could do that, and the brain maybe could do that. And is that the way we should envision place cells now, as being active in controlling everything that's happening around us that we can control? It's not representing things that move outside my control, right? Right?
Well,
Reassure me of that.
I mean, I think today we saw that place cells were still representing place, but it didn't necessarily have to be the place that the animal or person or organism is. It doesn't have to be the current sensory representation, but it could be thinking about that place, that I was there in the past and I need to remember that so that I can use that for information to get back there again. Or it could be thinking about the place I want to go to, right? Or consolidating the place I just learned about and sleep. So I think that the place cells were still representing a spatial location and place, but it just how it was representing that information in all these different contexts, not just necessarily I am in that space and this is a GPS signal, right?
So I guess my question is, only one of those things at a time? Or could I be thinking about something and have place cells firing, actually be somewhere and have place cells firing, be having a goal for a movement and having place cells firing, so there would be more than one place representation in the hippocampus at a time?
Actually, I think Albert's work really strengthens the idea of them being place cells, because, we've always had this issue, like not knowing, oh, well, they're these cells, they're firing, but how do you really know they mean to fire to space? Maybe it's some odor that was in that location or some sort of view. And there, I think this work to show that actually thinking about that place, because those animals really had to think about the place, including in novel ways that you wouldn't think, even like the one that you show where moving an object, because that's very different from what the animal does during the task. So to show that spatial thought drives the activity in the same way, I think it actually really strengthens the idea of place cells as spatial neurons.
I see, because you mean there are no local cues that they can experience at the remote place anymore because they're not there. Yeah, I think that's a very interesting way to think about it. Yeah, I think just generally, not in relation to the work we did specifically, but just if we are in a room, I mean, we have an egocentric idea of this object is to the right of us, this is to the left of us, and this is forward. But if we, let's say, exit that room and we have to recall where things are, then and we have to think about that independent of whether we enter the room from one side or the other, then we need to have a representation of that in allocentric space. And so that's where I think thinking about space remotely, but in allocentric terms, is where you would want to use this kind of non-local activity. So that's one way to do it. You could remember something was on the right, but if I exited this room and entered it from another side, that wouldn't be very useful to me. I should think about it being, it was near the, it was on that part of the wall near the screen, something like that. And that would be an absolute representation. So I think it's a way to think about memory. It's a way to recall memory in an absolute sense, spatial memory in an absolute sense, and then maybe to also manipulate it in ways that are potentially novel for creative purposes or for simulation and planning purposes.
I guess my question is there just one object in space? Or is there simultaneously many of them? So normally in an experiment, there's just one object in space. It's the animal whose place cells are being mapped. But potentially the animal could be paying attention to where it is and having place cells telling that, and also thinking about where it's going next. And I think you guys have seen that, and that's simultaneously being represented. And then, maybe even mentally imagining a movement and all of that stuff simultaneously. So there's more than one thing happening. I mean, it's not outrageous to think that in one part of the brain there could be more than one thing happening. I'm just thinking there isn't just one representation for space. And so that means all these different groups of neurons that represent different things are kind of coexisting in the same hippocampus.
Yeah, even the same neuron. So you can we know the hippocampus is important for episodic autobiographical memories, which is the what, Tolving that's the what, where, when. So you would expect that in that network, you would find neurons representing the information about all aspects of your experience, when something's happening, what's happening in the context where you are, and that would be integrated. And you can even see in individual neurons in the hippocampus that they will have these integrated codes where they'll have a spatial receptive field and represent a specific location, but they can use their firing rates to represent a specific cue, like a scent or a color or some non-spatial cue. But you have these dynamic firing patterns of using lots of different types of coding schemes to be able to conjunctively represent many aspects of the experience. So space is a really important component of your memory of what you're doing, but there's a lot of other pieces of information that's integrated with that space in the hippocampal code. Yeah.
I'll add that you, I think, brought up, can you represent the animal's current position and other things at the same time, like maybe this object in the task that Albert took? I suspect you can, just based on, we can see what we call the animal's representation of the local position and farther or non-local positions, but they're not literally at the same time. They're segregated by theta phase. So they're segregated tens of milliseconds apart. So they're in some senses at the same time, but not actually on top of each other.
Oh, so that's interesting. If I've got two representations in my place cell population, and they are happening on different phases of theta, then presumably I can keep them straight.
That's the theory.
So that means I can't have three.
Well, I think you probably could, but then maybe they get segregated on different theta cycles, for example.
Ah.
It's the theory that the theta phase is helping to segregate streams of information.
So there was an idea that the gamma phase was helping to sort out things that are happening throughout the theta cycle. What's the current status of that idea?
I mean, I think it's still
been
going.
abandoned.
Yeah. Yeah.
Supported. Yeah.
There are different frequencies of gamma at different theta phases. So they might be playing a similar role to the theta phase segregation in that a slow gamma, medium gamma, fast gamma, there are different theta phases and they are coordinated between different subregions of the hippocampus. So again, thought to segregate these different streams of information.
So all of those different things are held in a single map of space, and that keeps them in register, right? So that otherwise, if my imagined things that are in this room at this time have to be in the same spatial frame as real things that are in the room at the same time. But if I go into a different room, I guess the spatial mapping of my place cells will be altered. So that means that also my imagined things in that room will be altered. And that's fine because I'm in a new room. I'm in that room. But if I'm sitting in this room and I'm imagining, Albert gave us this little assignment to imagine ourselves walking around in our homes. And he told us to close our eyes to do this experiment. And I don't think closing my eyes was really absolutely necessary. I can imagine walking around in my home while I'm in this room. So now I have two completely different spatial frames somehow being represented in the same hippocampus. Is that hard to think about, or is that okay?
Well, one idea was that one of the ideas of the theta binding is that you would be in one location in one theta cycle, but the next theta cycle you could now be in a different location. I mean, that's 8 times per second. So you could flip pretty frequently without like being lost in space.
I mean, I would also argue, though, when I actually think about being someplace else, when I'm here, I'm not really paying attention to being here the same way. Like, maybe I know I'm still here, but the way that information gets processed or attended to at some point is very different. Whether that's within the hippocampus or downstream, I don't know. But I don't think it's quite the same as being like fully focused on being here.
So one thing that, I think that emerging from all the talks today, we got reminded of how more complicated the picture is that we were thinking only some years ago, because, all these things about, encoding where I am versus thinking and doing this mental exploration travel. The idea was, okay, there's different states of operation of the brain, mostly correlated with different local field potential states. And so you got the theta for exploration and the moment in which, what you're representing right now. And you have the sharp waves and ripples. It's just these flights of imagination, just so to speak. And it seems like the picture is getting way more complicated. And one thing I teach to my students is one of the most solid pieces of knowledge in neurophysiology of behaving animals is, going back to Van der Waals, is that there are these brain states, and the local field potentials, and they correlate with different behavioral states. And it is appealing to speculate that, okay, well, that is also extended to internal cognitive states. And so what I'm doing, again, being present in the moment here versus thinking of being somewhere else. And I think the picture is getting confused because there's not really this clear partitioning. So now we're starting seeing these mental exploration in theta, and so I think that's fascinating in a way, and in another way it's just like kind of you're just scratching your head because it's like, okay, well, now what? And there's even just right now, we're thinking about like different theta cycles or different phase of theta, completely different cognitive operations, essentially. I think that's, one thing that kind of emerged today from listening to all the talks, was on my mind, but which is kind of related, I guess the original question you asked about how all these things. Because there's one other thing we were thinking about. If there's all this idea of attractor networks, and so that holds a clamp, a variable, for example, a positional variable, okay, well, if now the attractor starts representing a remote space and a trajectory, remembering a trajectory that is somewhere else, how does it go back to where you are? Unless, okay, well, you can look, but what if you're in the dark? What if you're... And so where is that information? Where does that information go? How does it come back? And I mean, there is work by Laura and Frank, showing that some cells seems to keep encoding where you are. So there might be subgroups of cells, but I don't think it's completely resolved. So like it's interesting, right? Because we think about, okay, this is what holding this information, but then these things just do something else. So where has the information been written? How does it come back? And it's interesting, in this sense.
This is related to another theme that I was thinking about talking about, which is decoding.
Mm-hmm.
Because now all of you can record from a lot of hippocampal cells at the same time. This is maybe doesn't seem very new to you, but it's pretty new to me. and it changes everything, because now you're looking at a whole bunch of cells at the same moment, and you decode the whole group all at once to get a single spatial signal. And you've all had to work out a decoding algorithm for yourself. You share certain ones. And so this is automatically leading us into asking kind of the question about how is that information digested by the brain? And so have you learned something about how the brain has to decode this information from your own efforts at trying to decode it? And Cameron, you were talking about like iteratively learning the code.
Yeah, I mean, I guess I was thinking there are times where you can decode, and there's time where it doesn't make sense to try to use that activity to decode. So in that sense, it's not to say that you can always decode. In certain contexts, you can. So exactly like what you said earlier, while you're in this room, I might, if I had access and I could decode your activity, maybe I would see your thoughts jump over around locations in this room. But then if I applied the same decoder and you went into the other room, I would get nonsense, right?
Is it worrisome about how the brain is going to deal with the hippocampus?
Yeah, so then how's the brain know what to do with that?
It can't have a static decoder. It's got to have an adaptive decoder.
I am really intrigued by the work you showed where there's basically some ensembles that are very stable and over this novel experience, sleep, and then second novel experience, same track, second session. But then another set that are basically seem to be joining a different ensemble.
Right.
Yeah. So then from the point of view of a downstream neuron that's getting a bunch of inputs, what is it making of that? Like the stable one, okay, maybe it's like, yes, predictable. Right. But the one that changed, like, have you thought about what that means to a downstream neuron? Does it just...
Well, I mean, I guess I think about it in the context of like, how do people make sense of representational drift in general? And the main analogy I think of is that, you can't step into the same river twice, notion. So, it's something is different, like you're a different person. So in that sense, the space, like how we are representing the space is constantly changing, but because we're embedding, I mean, this is fundamentally like what Jill pointed out, like our system for representing our experience is declarative episodic memory. So there's these other components in the activity, including like the ensembles that are coactive, that are presumably representing different aspects like of time as well as space. So that doesn't answer like how it's decoded, but I guess I think
you answered my question, which is the second time in that same environment is not the same.
Right.
So some things are the same to a downstream neuron. It's getting some things that are the same and some things that are different, but that's actually important information. The difference matters.
Right.
If it's trying to sort the cells according to what kind of object they're representing, or, whether it's my location or something else, then it is going to be hard, because every time there's a cell changes membership with a group, it's gonna get drift farther and farther and farther away. So there's got to be some way to update the. You were kind of using a mechanism for updating. That's how you detected that a cell left one group and entered another.
Right.
I mean, I think what Cameron's showing is that, the assumption has to be, and I think the data shows it, that there aren't so many changes going on all at once. If that were happening, you wouldn't be able to, you could try it, but you wouldn't get out a clear answer like what you did. So it means that there are changes, but they are relatively slow, right? So that allows you to sort of, with bootstrap on what's left, right? And then do that for each cell separately. If it all of a sudden remapped completely, we could detect that by seeing that it's all nonsense now, but then we'd have to decode it a different way. But this seems like it's like a gradual change. Yeah.
Right. Which you would hope, like going back to the same location.
Yes, exactly. You want the river to be fresh. There. The river should be roughly the same, just not exactly the same.
Yeah.
But I think you pose a very difficult question for our community, and that is, how does the brain decode information? And so I think we do a really good job of recording activity patterns and describing them, and then saying what we think they mean and decoding them with algorithms. But do we know that the brain does that? And do I know that the downstream neurons, what they're reading or what they care about? And I think, a lot of the story is, do we know the full connectome about which neurons are connecting to who and what their information is? And the answer to that is no, not entirely. It's really hard to map on the functional cell types in these memory circuits to the actual connection patterns that they have. And so I would need to know, to determine the readout, like which cells send their axons, who's connecting, what's my connectome? And then I would need to know what kind of information's coming in and what are their temporal dynamics. So like the question that you pose is a hard one for us, because how does the downstream network decode the information that is coming in is, I think, a new domain for scientists of the next, that we're training, that are coming up, to really focus on. And I think that a lot of that, the tools are there and the information's coming in now that is needed. Large data sets, connectomes are getting figured out. There's lots of ways to look at activity, but I think how the brain really does decode information is a really important one that we probably can all hand wave here for a little bit, but I think is one that's hard to answer.
And let me add, not only decode, but how does he use it?
Yeah.
Because, a sore point of our field is that we still don't have a lot of evidence. It's like, okay, place cells, how they get used for some kind of behavioral output. There's rare
causal experiments are there,
experiments,
but it's not
and so the literature is a little, here and there, and so there are some examples. As a matter of fact, Isabel Muzio, who was one of our formerly owned faculty here, she has studies in which she has some behavioral readout of remapping, like with the animal making mistakes based on which map is recollecting. It's kind of not frequent. You see that in the literature. So there is also that. And people ask, and then you don't have a lot to give them, but of course, if you've been studying this phenomenon for very long, there is encoding of position, and then you have like all these replay of trajectories, and so there's something being used that's a cognitive level, but yeah, you would then the behavioral readout, because the behavioral readout is important, or some other cognitive operation readout, because otherwise, if we're only talking about decoding, I would just always go back to the homunculus. There is some little person there trying to read out, and okay, but that's gonna become circular with the brain, right? So it'd be better to say, okay, well, how does that affect something else? And then that something else is a little elusive sometimes. That's kind of what my take on it.
I mean, the Muzio example is nice. Also, there's the Lavillon experiment that everyone was really surprised by. The one where they rewarded when the animal was sleeping. They rewarded the animal while they were sleeping when a place cell fired, and then when the animal woke up, they would run to that spot because it was associated. You associate reward with that place cell sort of in an offline period. And then there's the Robinson-Heuser recent study where they did holographic stimulation and activated a subset of place cells that were active in a reward zone. When they did it, activated it in a different area, then the animal licked as if they thought they were at the reward zone. So I think those are like the direct causal experiments I can think of. But there are few, right? In terms of really reading the specific code, I think it's just difficult. It's a non-topographic code.
I think that's an important comment, though, that testing the causal, there needs to be a step that we causally test some of the prominent hypotheses that there are in the field, right? And for example, in my talk, and when you see these beautiful codes that are playing exactly where the animal's going to the next choice, and you see this beautiful correlation between that representation and the animal's next choice, but then when you disrupt that and do a causal experiment, it doesn't change the behavior at all, right? It says to you, oh, well, maybe that's something else. And as you mentioned, those awake ripples seem to be more tightly correlated with the consolidation during sleep, right? So I think causal experiments are really important to just shift the descriptions or what you're thinking about in terms what we describe in the data and what we apply it to in our theories need to be tested at some point so that we can then transition appropriately to what their meanings are, what they're serving in terms of
I mean, the planning function is also, evidence that these places are potentially guiding the behavior.
By the way, if you're listening, Isabel, we miss you.
I would add, I mean, the behavior causal manipulation and a behavior readout are phenomenal and really hard, but also even just like the next steps in the circuitry, what's the next computation and the computation after that, all the way out to the motor behavior, that's severely lacking. And I don't even have a good mental model of what's going on through those steps.
So before we quit, I'd just like to comment on something. I mean, neuroscientists are used to thinking about events in the brain causing experiences, sensory stimuli, mental events. But all day today, I've been just marveling about the way you have reversed the causation. Mental events are now causing firing in the brain, patterns of firing in the brain, not the kind of neuroscience I grew up with. And it's natural. It just happens naturally, comes right out of your experiments and your results. And it fuzzes the line between what causes what in our lives and in the brain. And I don't know, have you had any, because you deal with this all the time, does it have any insights about that? Where should we go with that? Should we pursue that idea?
I mean, I think we should. I think part of probably what we're all inspired by is we want to understand thinking, right? Not just a response to a sensory input or the trigger of a motor action, but we want to understand thought.
But we are also focused on the hippocampus, which is flexible, right? And I think that it allows for this type of inquiry. If we were looking maybe motor cortex or premotor, maybe, in that sense, it couldn't be driven by imagination or thought alone.
But isn't that all why we studied the hippocampus?
Yes.
Yeah. I mean, I think the motor system, you are also imagining actions. I mean, that's how I think the BCI that has that uses intuitive, sort of the naturalistic coding that is very closely related to what fired when let's say the animal actually moved. Let's say their arm or push buttons to move a cursor to a certain spot. If you use that code, then when you go to the BCI mode, they are readily able to generate those patterns because that is what they're naturally
they don't
doing.
even have to move.
Right. They are thinking, right?
Yeah,
So, yeah, I guess, ultimately we're complex machines and you can go back and back and back and back in terms of causation, and there'll be some very complex or hard to pinpoint actual cause. You heard someone say something, and then 5 hours later, you're reminded of it, and then it causes you to drive to this place versus another place. Yes, the cause was that plus all of your other memories from your whole life. So
that motor experiment
hard for an
that
animal
you just
experiment.
mentioned is like really famous one, I think, where a monkey was operating a manipulandum to operate one of those grabbing games where you never win, but the monkey could win sometimes.
Yeah.
And they're recording from the motor cortex that they think are the neurons that are driving the arm. And once they figure out the relationship between the motor cortex's firing and the movement of the arm, they quit connecting that grabber arm to the manipulandum and just connect it straight to the neurons. And at that point, you would go, my expectation is fine. It's still going to work. The monkey's still going to move his arm because the neurons are still firing. And that's what makes the monkey move his arm. And everything's going to happen. You're not going to see any difference. But there was a difference. The monkey quit moving his arm.
They've taken it another step further recently, from my understanding, is now they actually, because let's say initially, two neurons that co-fired in the same way during the task. Now they've made the animals' reward depend on them causing these two neurons to be like uncorrelated firing. And it seems that so they can challenge the animal by basically having new rules for driving the cursors that are, anti-correlated with the natural movement, and animals are able to accomplish this. So in that sense, there is a remarkable plasticity. I guess it's really the end result that somehow can be brought about.
Motor cortex connection to the muscles is.
Although it seems like some neurons are more easy to decorrelate than others, is what they're exploring. Right. But
yeah.
Well, thank you, everybody. It was a great symposium, and it was fun talking to you. Really appreciate you.
Yeah, thank you. We really great symposium today.
Yeah.
Thank you. Thank you, Francesco.
Yeah. Thank you for coming.
I learned a lot.
Thank you for coming.
This has been Neuroscientists Talk Shop.